I'm Dr. Joe Flaherty, and I know biology. I'm very fortunate in that I've had graduate training in the realm of genomics and bioinformatics. I brought these learning experiences with me to Coker College in that I specifically trained students here to utilize the most recently developed tools to analyze genomes of organisms in the process of gene discovery. To that end, students have been actively trained to use bioinformatic tools to compare genomes from specific fungi. Part of the research support that we have received, we have been able to purchase relatively expensive equipment to analyze gene expression. Why is this important? Well, part of the gene discovery process involves identifying genes that are turned on under certain conditions. So we use a guilt by association approach to identify important genes to a particular process. And in doing so, we culture our fungus in two conditions. One, in conditions where the fungus does not produce canidia. It senses that there's enough food in the environment. It does not need to produce the survival structures. We compare the genes that are expressed from under those environmental conditions, compare that to environmental conditions that stimulate spore production. And in doing so, we can subtract the genes that are expressed that are in common and find the uniquely expressed genes under the situation that stimulates production of spores. Once you identify those specific candidate genes, now you can look at their expression individually and validate whether or not they are only turned on during this particular developmental process. This is a step in a direction that will ultimately lead us to experiments that test the hypothesis that these particular candidate genes are directly involved in that particular trait, which in this case is the developmental decision to produce spores.